All right, hi everyone. Today we will be continuing with our unit on genetics with Mendel and the gene idea. Please make sure that you have your notebooks open and you are continuing with notes wherever you left off. And we left off with learning how to set up and solve Punnett squares and applying some genetics terminology as well. So let's run through this practice all together just to make sure that we understand what's going on. When told to solve a Punnett square, you will be given a character, and sometimes you're given a letter. Sometimes you have to come up with a letter on your own. And in that case, it doesn't really matter what the letter is. And you would need to know some information, like what is the dominant trait and what is the recessive trait. So in this case, it's flower color. We'll use the letter P. Dominant is purple, recessive is white. We also need to know a little bit about the parents. So if parent one is heterozygous, remember this word means the two alleles are different. One is dominant and one is recessive. So this parent genotype would be big P, little p. We need to know about the other parent as well. If they are homozygous recessive, homozygous means they have two copies of the same allele. Recessive means they have the recessive one. So they would be little p, little p. Now to set up this Punnett square, let me write on here. Okay. We need to create the parent gametes, which just means we need to separate the two alleles and put them on the side of the Punnett square like this. I'm going to make it very obvious that one is big P and the other one is little p. And then up here, very obvious, little p, little p. Okay. So these are the two gametes of the parent two. These are the two gametes of parent one. Okay, remember that this is applying Mendel's law of segregation, that alleles separate during gamete formation. And we know that because gametes are haploid, they come from a diploid parent, so we must split apart those two alleles. Oops, did that wrong. Sorry, I can't multitask. Big P, little p, little p, little p, little p, little p. Okay, excuse my writing. This is hard to do on a mouse pad, but we're going to do our best. Okay, great. And hopefully we understand and can follow this. This is just kind of like multiplication. We stick the big P with the little p, little p, little p, little p, little p, and we end up with this Punnett square. Now, with this, we can answer all sorts of questions, like questions about genotypes or questions about phenotypes. So this flower here would be purple, this one would be purple, this one would be white, and this one would be white. So we can answer questions that are asking for a ratio, a fraction, or a percentage. Okay, and you got practice with this on your most recent homework, and you'll get some more practice with this on that first page of your genetics work packet. Okay. How do I clear this? Clear. Clear. All drugs. All right. Cool. Let's continue then. Now, sometimes we don't actually know the genotype of the parent, so we can do a Punnett square, but just kind of backwards. And how you would do this would be by using a test cross. Okay. Now, this works best with plants and things that you can purposely breed together, preferably quickly, so that you can see the offspring and then kind of backtrack and figure out the unknown parent. Okay. Um, so, for a test cross, you need to cross the unknown parent with a parent that is homozygous recessive. We would know that this parent is homozygous recessive because they are displaying the recessive phenotype, right? This is a white flower. The only option for its genotype is little p, little p. This is a purple flower. So actually, we don't know if this flower, if this parent genotype is heterozygous or homozygous dominant, right? This one could be big P or it could be little p. We know that there's one big P at least, but we're not sure about the other one. So we could take this purple flower, cross it with this white flower, allow them to create new flowers, and then observe the offspring. So in this case, we would have these two combinations for sure. But then if the parent were big P, we would end up with this child and this child. So we would expect one, two, three, 4 out of 4, or 100% of the offspring to be purple. Okay, and these are plants, so when they produce offspring, they can produce a bunch of offspring, which makes plants favorable in using test crosses for. So if all of the offspring turn out to be purple, we can be pretty confident that this parent's genotype is big P, big P. Now let me use a different color. Let's say instead this parent were big P, little p. Well, these two would be the same, but now here we would have little p, and here we would have little p. So we would expect 50% of the offspring to be purple and 50% of the offspring, 1, 2 out of 4, to be white. So if you cross these two, observe all of the resulting offspring, all of the resulting flowers, and there are some that are purple and some that are white, well, then it has to be this genotype here. So this is how you would solve for a parent's genotype if you're not sure what it is. Run a test cross. Okay, this is just doing a Punnett square just a little bit backwards. All right. So all of these crosses, these Punnett squares, and then even these test crosses are things that Mendel actually did. And he truly looked at the answers to questions like this, resulting genotype ratios and phenotype ratios. And please just keep in mind that these two ratios are not always the same. So we have one homozygous dominant to two heterozygous to one homozygous recessive. That's a 1 to 2 to 1 ratio, and this is the way you should write it. Okay, notice that the genotype is written beside the number because we want to know which is which. Now, sometimes the genotype and phenotype ratios are the same, but sometimes they are different, right? So in this case, these two options produce purple flowers, and this option produces a white flower. So our phenotype ratio is 3 purple to 1 white. Okay. So Mendel really did these sorts of things. He really did these crosses with parents that he knew and observed the ratios and percentages. And he also did those test crosses to backtrack it using ratios basically to figure out the parents. Okay. Now, sometimes there's a little bit more complexity that we want to keep track of. Sometimes we care about looking at more than one gene at a time. And if we want to do that, we need to remember Mendel's law of independent assortment, which states alleles of a pair segregate independently of other allele pairs during gamete formation. So we know that alleles separate during meiosis, gamete formation, so that each gamete ends up being haploid. And we know that this happens randomly, and we applied that principle on our Punnett squares. But what is also true is that alleles for different genes segregate independently of each other. In other words, genes don't care what other genes are doing. Now, something to keep in mind is this is only observed when genes are far apart from each other, on the same chromosome or on completely different chromosomes. And the reason we need to keep this in mind is because if they happen to be close together, they might actually be inherited together during crossing over. So remember this process from meiosis where they just kind of swap some genetic information. If two genes are close together, then it's decently likely that they will be crossed over together as well. Opposed to genes that are far apart, it is a lot less likely that they would be kind of linked together. Okay, so we can usually assume that two genes will not be inherited together. They will be completely random of each other. But there are cases where they are linked, and we'll talk about that. So let's assume that the genes are far enough apart that they will not be carried over together during crossing over. Then we would do what's called a dihybrid cross. This is just a double Punnett square. So what we have been doing are called monohybrid crosses, where we are tracking one character. Okay, and we're looking at what happens with just one character that's randomly separated and randomly combines with another allele. For a dihybrid cross, we're looking at two things at the same time and trying to come up with all of the possible combinations. So, for example, in humans, we might use a monohybrid cross, a Punnett square, to predict eye color. We would use a dihybrid cross if we wanted to predict eye color and hair color at the same time. Assuming that eye color and hair color are far apart from each other and they will not be linked together, we could come up with all sorts of combinations, right? Blue eyes and brown hair, blue eyes and blonde hair, brown eyes and blonde hair, brown eyes and brown hair. So there's kind of like double combinations. And that's exactly what we will use a dihybrid cross for. So let's take a look at this dihybrid cross. Okay, this is a dihybrid cross in its most complicated form. And this is a true dihybrid, meaning two hybrids. So two parents who are double heterozygous. Heterozygous for two different characters. So Mendel actually looked at this when he was self-pollinating his F1 generation. And if he got a plant that was double heterozygous, big Y, big R, little r, right? Heterozygous for the Y and heterozygous for the R, double heterozygous, and he crossed it with itself, then we would end up with two double heterozygous things. Okay? All that is to say that this is as complicated as the dihybrid cross will get. Okay? Now, solving a dihybrid cross is just like solving a monohybrid cross, except for there's one extra step in the beginning. And that is the step where we have to distribute. And what we mean by distribute is to determine all the potential allele combinations, meaning all of the ways the Ys compare with the Rs. This is because of the law of segregation, right? And the law of the independent assortment. The Y gene doesn't care what the R gene is doing. So Y can combine with R in all sorts of ways. So we need to predict all of the possible gametes from the parents. And we're going to do that with foiling or distributing, like from math. Hopefully you remember foiling, but I'll show you anyways. Once you do that, you'll set up your Punnett square just like normal, and then you can solve. All right, let me show you what I mean by foiling. Now, in this picture here, please don't get this confused. This is the P generation crossing to get the F1 generation. This giant Punnett square here, this dihybrid cross, is a cross of the F1 generation with itself. It doesn't have anything to do with these two parents. So go ahead and ignore those. That's just showing how we got here. So we have big Y times, or big Y, big R, little r times itself. Whoa, this is challenging to write on. Okay, like that. That's the cross that this Punnett square is. Okay, so this first step is to distribute. Figure out all the possible gametes. Here's how we do that. Big Y distribute to big R. Here's one gamete. Little y, big R. Big Y, little r. Little y, little r. I just distributed. We could do the same thing. We would do the same thing for the second parent. Big Y, big R. Big or little y, big R. Big Y, little r. Little y, little r. Like that. And that's how you set up your Punnett square. Then you go through and you solve. The same way that we did before, keep the y's together and keep the r's together. So big Y, big Y. Big R, big R. Let me do a different color. think. Big Y, little y, big R, big R. Show you one more. Then I think we got the hang of it. Purple. Big Y, big Y, big R, little r. Okay, that's what we're doing in all of these squares. Now it's just a 4 by 4, not a 2 by 2. This is going to be some more time consuming. And then the next part is to determine the phenotypes, depending on what the question is asking you. But for dihybrid crosses, we usually want to figure out the phenotype ratio because we're kind of interested in the possibilities of these two genes and how they can combine together. So how do we figure out a phenotype ratio? Well, we have to go through and figure out the phenotype of each box. And let's make a little key here. So in this Punnett square and what it's, I don't think it even tells you. So let me tell you, we're tracking pea color and pea shape, like Mendel's pea pods, right? Or Mendel's peas of the pea pods. So for pea color, it can be yellow, yellow, or green. Okay. And yellow is dominant. Big Y, green is recessive, little y. We're also tracking pea shape. There are round peas. Okay. And that one is dominant, big R. And there are wrinkled peas. Ooh. Okay, that's wrinkled. And that's recessive, little r. Okay. So now that we have this key, we could go through and determine the phenotype of um, each P from this result here. So let's do that, or let's at least start it and then I'll let you figure it out from there. Okay, let's look for yellow round peas first. So here's how I like to do this. I like to color code things. So I like to go through each box and determine its phenotype, but keep track of it with like a 4 color key basically. So I'm gonna look for yellow round peas first. Okay, yellow round peas first. Now remember there's multiple genotype possibilities for yellow round peas because both of these are dominant. So we're looking for homozygous dominant or heterozygous dominant and any combination of those. So let's look at this first pea. This has big Y, so it would be yellow, and big R, so it would be round. So this one here would fit my phenotype here of yellow round peas. This one would also be yellow and round. So would this one, so would this one, so would this one. This one would not. This one would be, not this one. Yes to this one, yes to this one. No, no, yes, no, no, no. And if I went through and counted, that's 1, 2, 3, 4, 5, 6, 7, 8, 9 yellow round peas, which is what this says right here too. And then we can go through and do the same thing for the others. Let's use the color green. So now let's look for green round peas. Okay, now green is recessive. So we're looking for 2 little y's and then a combination of heterozygous or homozygous dominant for shape. Now we've already counted these boxes. I color coded it, so we're good there. We're good here. Okay, this one would be yellow and round or green and round, right? This one would be green and round. Not this one. No. This one. No, no. So I end up with 3 green round peas. Okay, let's keep going. We'll use dark blue now for, how about yellow and wrinkled peas? Yellow and wrinkled peas. So I'm looking for the dominant yellow again, and then I'm looking for the recessive shape. Okay, these ones I've already marked with a different color, so not those. This one would be yellow and wrinkled. This one would be yellow and wrinkled. This one would also be yellow and wrinkled. 3. And then one more. Let's use orange for now green and wrinkled. This is a double recessive thing. All my boxes have already been marked, so that just leaves me with this one here, like that. So the resulting phenotype ratio is 9 to 3 to 3 to 1. And this ratio is like the golden ratio of a true dihybrid cross. Every single time you cross something that is double heterozygous with something that is also double heterozygous, you will always get this ratio. If the parents are not both double heterozygous, you will not get this ratio. But this is the ratio Mendel found, and it is the ratio that will always be for this double heterozygous parent. Okay? Like I said, this is as complicated as a dihybrid cross gets because this has the most unique combinations. Okay, so hopefully you were able to follow along with that. Clear all drawings and moving on. All right, let's practice with a different set of parents that have different genotypes. Ignore this. I don't know why it's there already. Let's say I have these two parents here. I need to figure out the possible gametes first. Okay, I need to do the distributing thing. Let's use black. Actually, let's use blue. So this is parent one, and I need to figure out all of their possible gametes. So I distribute big C, big P, big C, big P. Okay, I already have that one. Big C, big P, big C, big P. So it just so happens that this parent is only big C, big P. So I'm going to write that on all 4 lines. Now let's look at the other parent and find its possible gametes. Little c, big P, little c, big P, little c, little p, little c, little p. So now this case, I have 2 possible parent gametes. So I write that here, and I'm just going to repeat it to fill up all 4 spots. Can I type? Oh, I can type. Let's do this. All right, now you would just go through and cross. Big C, little c, big P, oops, big P, big P. Keep it all together like that. Okay? Keep going. Big C, little c, big P, big P. Big C, little c, big P, big P. Big C, little c, big C, big P, big P. Okay? Why don't you pause the video at this point in time and try to fill in the rest on your own. All right. And go ahead and check your answers. Did we get this? Hopefully we were able to follow along and complete this Punnett square. Now, what you probably realized while doing this example was that it became very repetitive. And that's because it is very repetitive. Sometimes that happens. And if we knew more information about phenotypes, we could go through and assign the phenotypes to all of these genotypes, but we weren't given that information, so we don't know. And what we would find if we were capable of doing that is, well, this one would look like this one, and it would look like this one and this one. And actually, if we went through all of them, they would look exactly the same because they all have one dominant of each character. So this is an example of the parents having different genotypes, and therefore the resulting phenotype ratio is different. Okay, So that's a dihybrid cross. You have two examples of these in your genetic work packet. Warning, that worksheet is not one that I made and it does have some typos on it. So please ignore the typos and just pay attention to the given genotypes. And what I want you to get out of that worksheet is to learn how to create the gametes, do the distributing, and then solve the Punnett square, solve the dihybrid cross. Okay, that's the biggest takeaway. Please do your best to answer the questions that are on there as well. But really, I just want to make sure that we know how to set up and solve this. Okay. All right. Now, we said that that dihybrid cross works for genes that are going to separate independently of each other. They're far away on a chromosome. They're on completely different chromosomes. They're not going to be inherited together. But actually, sometimes there are genes that are inherited together. Those are called linked genes. Linked genes are found close together on the same chromosome, and they tend to be inherited together. They tend to be linked. This is because when crossing over happens, they probably won't be separated because they are so close together. So that arm of the chromosome that sticks out and swaps will swap these two genes together because they're just so close together. So that's what a linked gene is. And if we look at this picture here, this says purple flowers and long pollen are inherited together. Red flowers and round pollen are also inherited together. And we can see this because when we look at the ratio of how many purple long flowers there are compared to purple round flowers, it's like a very extreme difference. So it is most likely that the purple gene and the long pollen gene are like right next to each other, and only in these rare cases do they get split up during crossing over. The same is true with the red gene and the round pollen gene. So that's what we mean by linked genes. Now, notice here there are still some purple round and red long flowers because there are some odd things that happen during crossing over. It is just less likely that linked genes will be separated. Okay, so it is still possible, just less likely. Now, we know this information because mapping genes is a thing, and these genes that are found together on the same chromosome tend to be inherited together. And we actually know the likelihood based on mapping genes and creating recombinant frequencies. A recombinant frequency is the chance of two genes being recombined or separated from each other and recombined elsewhere. So smart people who know how to figure out the genetic sequence of something or the complete genome even can identify genes and their position on a chromosome. And then they can also kind of map this out and see how far away those two are from each other, basically by looking at the amount of nucleotides in the middle. Okay, so even if you get like the complete genome, you can piece together one gene is here, the another one is here, and on and on and on. I realize I'm making hand motions and you cannot see that, and I'm sorry, that's okay. And we can review this too later, but here's a map. So let's look at what's happening in this map here. We have three genes, G, C, and I, or L. I think that's an L. And someone knows where they exist on a chromosome. So the G gene is here, C is here, L is here. And because we know the length of the genes and we know how many nucleotides are in between them, we also know the recombinant frequency. Between G and L, based on some math, there is a 17% chance that they will be recombined, meaning there's a 17% chance that they will be separated during crossing over. G and C have a 9% chance, and C and L have a 9.5% chance of being recombined, a chance of being separated. Now, hopefully this makes sense. G and C are very close together, right? They're super duper close together. So it is less likely that they will be separated during crossing over. Same with C and L. But G and L are farther apart, so it is more likely that they will be separated. Okay, so let's answer this question. Which genes are most likely to be separated during crossing over? It should be the one with the highest recombinant frequency, the two genes that are farthest apart, so G and L. It is still possible for G and C and L to be separated. It is just less likely. So mapping genes has helped us greatly with knowing a little bit more about the genetics of linked genes and predicting what will happen. Hopefully now these frequencies help explain this picture here of how it is still possible to end up with these options, but it is more likely to end up with these options. So that's what we mean by linked genes. A standard dihybrid cross would not work so well as a true predictor for these genes that are found close together. Okay. All right. I have a little bit of more math for you because sometimes we care about more than just two genes. And if we try to make like a tri-hybrid cross, that becomes really confusing. And then even sometimes we want to look at like 10 genes, 20 genes, and it becomes too big and too challenging. So instead, we can just use some math rules. We're going to use the rule of multiplication and the rule of addition. The rule of multiplication says that if two independent events happen and we want both of them to happen, we're going to find the probability of the two happening independently, their own probability, and we're going to multiply them together. So if we want the probability of one thing and a second thing, find their individual probabilities and multiply them. The rule of addition says that if we want two things, either or to happen, then we find the probability of those two events happening separately, add them together, and then that's the overall probability of either or happening. Okay, let's look at an example. Let's use a dice, and a dice has 6 sides, and if it is a fair dice, then it has an equal chance of landing on any of the sides. So let's say you're going to roll the dice, and you want the probability of rolling a 6, this situation here. The probability of that is a 1 of 6 chance. Okay, easy. But now if we want the probability of rolling 2 6s, so now I want to roll the dice and I want to get a 6. And then I want to roll the dice a second time and I also want a 6. I want 2 6s. What would the probability of that be? Try to work this math out real quick. Okay. Well, the probability of rolling 1 6 is 1 over 6. The probability of rolling a 1 6 again is also 1 out of 6. And because I want a 6 and a 6, I'm going to use the rule of multiplication. So I'm just going to multiply these two fractions. If you have forgotten how to multiply fractions, don't worry. You just multiply the numerators. So 1 times 1 is 1. Then you multiply the denominators. 6 times 6 is 36. And it's as easy as that. The denominators do not need to be the same. You just multiply whatever it is. So you have a 1 in 36 chance of rolling 2 6s. Now let's think of a different situation. What is the probability you have a dice and you want either a 6 or a 2? I'm going to roll one dice and I don't care if it's a 6 or a 2. We're going to instead use the rule of addition. The probability of rolling a 6, we already established, was 1 over 6. The probability of getting a 2 is also 1 over 6. Because I want a 6 or a 2, I'm just going to add the fractions. If you forgot how to add fractions, here you go. If the denominators are the same, you simply add the numerators. So 6s are the same already, so I just need to add 1 plus 1. 1 plus 1 equals 2. 2 over 6 simplifies to 1 over 3. Okay? If the denominators are not the same, then you need to find the least common denominator and convert and make the denominators the same, and then you can add it very easily. But in this case, they're already the same, so no problem. Now let's think about these two answers here. Which one is more probable? 1 out of 36 or 1 out of 3? Okay, hopefully you said 1 out of 3 is more probable. Think about it. Let's say you are in a class of 36 people. The chances of me randomly selecting your prayer card out of 36 people is decently low. If there was only 3 people, then your chance is a lot higher. And actually, this is well observed in my classes. I have a class of 9 students, and they all pray all the time. It's very likely that I'm going to pull their card on any given day. Versus your class has 22 students, you're my largest class. You probably are not praying every day. You have a lesser chance of being chosen. Okay, so this is a smaller number than this number. Let's think about these two scenarios. This one, I'm really picky. I want this and that. It must be both. That's super picky. That's less likely. In this case, I'm like, yeah, I don't really care, 6 or 2. I'm less picky, so it's more likely. Okay? Now, how does this apply to genetics? Well, like I mentioned, once we get beyond 2 characters, trying to set up a singular Punnett square is challenging. But what's not challenging is splitting it into several little Punnett squares, finding the probability of individual things happening, and then applying either the rule of multiplication or addition. So let's look at this example here. I have these 2 parents with their genotypes, and I want the probability of this offspring here. Big A, big B, big B, big C, big C. Now, to set up a giant Punnett square and find this genotype, I suppose is possible, but it's going to get complicated. So what I'm instead going to do is set up, let me get the pen, 3 separate Punnett squares. I'm going to set up big A, right here, big A, little a, and solve. Big A, big A, big A, big A, big A, little a, big A, little a. Then I'm going to set up my second Punnett square. Big B, little b, big B, little b, big B, little b, big B, little b, and solve. Big B, big B, big B, little b, big B, little b, little b, little b. Okay, cool. And then last one, big C, big C, big C, little c, and solve. Big C, big C, big C, big C, big C, little c, big C, little c. Okay, cool. 3 easy Punnett squares. Now what I want is to find the probability of this offspring, and all I'm going to do is find their individual probabilities. So what is the probability of getting big A, big A? Okay, 1 out of 2. 1, 2 out of 4 equals 1 over 2. What is the probability of getting big B, big B? 1 out of 4. What is the probability of getting big C, big C? 1, 2 out of 4, 1 half. Now I have their individual probabilities. Now I need to decide, am I going to use the law of multiplication or the law of addition? Well, because I want this complete genotype, I want this and this and this. And is multiplication. So I'm going to multiply. 1 times 1 times 1 equals 1 over 2 times 4 is 8 times 2 is 16. So the probability of getting big A, big A, big B, big B, big C is 1 over 16. Okay? Let me give you an example of when we would use the other rule. Let's look at the middle Punnett square here. If I wanted the probability of getting big B, or little b, then I need to find their individual probabilities. Big B is 1 out of 4. Big B, little b is 2 out of 4. I would use the law of addition because I want this or that. My denominators are the same, so I leave it. My numerators, I just add. 1 plus 2 is 3. So there is a 3 out of 4 chance that I get big B, or big B, little b. And look at that. 3 out of my 4 boxes have those genotypes. So that is how we would apply those two rules of probability. Okay. All right, I think at this point we are Punnett square experts. So let's learn about one more tool where we actually still do apply Punnett squares, but we're going to visualize things in a little bit of a different way. This is called a pedigree. Let me use laser pointer. This pedigree here is a family tree. Okay, pedigrees are family trees, but there are some rules to follow so that they can be universally used in science. First of all, we need separate symbols for males versus females. Males are always square, females are always circles. This is important. Some pedigrees have a key and it tells you. Other pedigrees do not have a key, so you just need to know that squares are male and circles are female. Okay, sorry. I wanted to check that I was still recording. So please remember that squares are male and circles are females. Now, another reason it's important to differentiate between male and female is because we'll see that sometimes things only apply to females and sometimes it only applies to males. So it's important to know which is which. Now, also, like in family trees, there are lines connecting the circles and squares. Horizontal lines indicate parents. So here is the mom and the dad that are married. If the horizontal lines are the parents, then the vertical lines are the children. This mom and dad have 1, 2, 3 children. So there's a vertical line connecting to 3. This mom and dad are married and have 1 child. This mom and dad are married but do not have any children yet. As you can see, there are 3 different generations. 1, 2, 3. Each level is a new generation. So answer this just maybe in your head. How many children do the parents of the first generation have? Parents of the first generation have 1, 2, 3 children. Of those 3 children, how many are married? 2. 1, 2. The one that is not married, are they a male or a female? Female. How many grandchildren would they have? 1. Okay, so that's how we would read this. Now, the last part is some of the circles and squares are shaded and others are not. What we are shading in is whatever trait we are tracking for that pedigree. Now, we're looking at phenotypes here because phenotypes we usually know, or it's very visual. We can see them, so it's easy to track. So let's say, for example, this pedigree was for tracking blue eyes in a family. Every person who has blue eyes, which would be very easy to tell, you just have to look at their faces, you would shade in that family member. So this female has blue eyes, this male has blue eyes, this female has blue eyes. That means this male has brown eyes, this female has brown eyes, brown eyes, brown eyes. Now this one here says it's a carrier. What that means is that this person does not have blue eyes, but they carry the gene for blue eyes. They carry the allele. They would be heterozygous. Let's look at an example of a pedigree. We're going to use this example here with hairline. Hairline is recessive, or straight hairline is recessive. So Kristen here has a straight hairline versus the dominant widow's peak. Lori has a widow's peak. Straight hairline is recessive. And if we want to track that straight hairline going through this family, then the first thing that we want to do is fill in all of the genotypes of people who have a straight hairline. If straight hairline is recessive, there's only one option for their genotype. That is little h, little h. So Charles is shaded in, meaning he has the straight hairline. So his genotype is little h, little h. Everyone else who is shaded in, Frank, Gary, Juliana, and Kristen, are also all little h, little h. So we have now solved a lot of people's genotype, which is great. Now for widow's peak, which is dominant, this one is a little bit more challenging to figure out because now there are two possible genotypes for someone who has a widow's peak. Those genotypes are big H, little h, or big H, big H. It's dominant. So now this becomes like a puzzle to figure out as many genotypes as possible. Let's think about Debbie. There are two possibilities for Debbie. But Debbie has a child who has the recessive hairline. So therefore, Debbie must be heterozygous because Juliana needed to receive one H from Charles, one little h, but she also needs to receive a second little h from Debbie. So Debbie must be heterozygous. And if you're not sure, do a Punnett square. Let me do one real quick just to show you. Okay. If I wanted to see, here's Charles, little h, little h. And let's say I wanted to see if Debbie was homozygous dominant. I would go through and fill this out. And I would see that it is impossible to get Juliana's genotype. So I would rule out big H, right? These are all heterozygous. So then I could instead, let me just write over this, maybe in blue. I would instead try big H, little h. Okay, these would be the same, but now instead I would have little h, little h, little h, little h. Now Juliana's genotype is possible right here. So I can conclude that Debbie must be heterozygous. Okay, so we can use Punnett squares to help solve pedigrees. Now we can finish with Isabel here. Isabel must also be heterozygous because she's going to receive one little h from her dad, Charles. So she must be heterozygous. Now we've solved this complete family. Let's go over and look at Al and Beth and their family. Al and Beth have two children who are double little h, so homozygous recessive. So they must both be heterozygous so that they can pass along each one little h and give it to their children. Now they must each have one big H because they themselves have a widow's peak. Now we can also go through and figure out Evelyn and Henry, their other children. Now, if we do a Punnett square of these two, which I would encourage you to do this on the side and see, we would see that it's possible for Evelyn to be big H, big H, little h, little h, big H, or she could be big H, big H, big H, big H. And we're not exactly sure. So that's the other thing about pedigrees. Sometimes we don't actually know exactly, and that's okay. You're going to end up probably with some unknowns. That's fine. Now, maybe one day we could know the genotype of Evelyn for certain if she gets married and has children of her own. Now, for Henry, we have that same predicament, but Henry is married and has children, so we can probably narrow it down. Okay, now Isabel, his wife, is heterozygous, and they have a child, Kristen, who's homozygous recessive. So Henry must also be heterozygous to end up with that option. Then lastly, we have Lori. And if we do a cross of Henry and Isabel, we would see that Lori is in the same boat as Evelyn. Not sure exactly, but if she were to get married and have kids one day, then maybe we could know for sure. And that's how you do a pedigree. You won't be able to determine every genotype, but we could at least figure out a lot of them. It's kind of like just solving a puzzle. Now, this is a pedigree that did not go through and half shade the carriers. That's only done sometimes. So if it were one, we would half shade in Al, Beth, Debbie, Isabel, Henry. Can't shade in Evelyn or Lori because we're not sure. So if we really wanted to see who was a carrier, it would be possible to shade in half of the circle or square for all of those who are heterozygous. But this is hairline, and we don't really care what someone's hairline is. So it doesn't really matter who is a carrier and who is not. This becomes more important in traits or genes that cause disease or disorder. And many diseases or disorders are truly caused by just one single gene, making them of more importance for tracking via a pedigree. We would want to know who is affected by the disease or disorder. We would also want to know who a carrier would be. This is a lot more concerning. So this is kind of the more practical application of using a pedigree. Hairline is fun and all, but it does not affect somebody's life like a disease or a disorder would. Now, a lot of dominant disorders are more obvious and known, and recessive disorders become a little bit more tricky because they can kind of pop up at random. Not at random, but they can kind of pop up differently because they are recessive. So parents could be carriers, not even know that they have the disease, and the child could end up inheriting two bad copies of a gene and end up with a recessive disorder. So the carriers that are phenotypically normal could carry the allele for a disorder. So if we look at this family tree over here, we have two carrier parents. They might not even know that they're carriers because they're totally healthy. And they have the possibility of giving two good copies of the gene to one child, or one bad copy with one good copy, 2 out of 4 chance, or two bad copies, 1 out of 4 chance. Now, this is where parents might make a decision. They might say, okay, my odds are pretty good that my child will be healthy. I'm going to go ahead and have children. Or they might say, you know what, this 25% chance is really concerning to me. I don't want to take that risk. Or they might say, wow, this 50% chance that my kids will be fine, but now they need to worry about their kids, that might be too high of a risk. So these pedigrees are actually really important tools for parents to make informed decisions. Okay. And these types of diseases and disorders, as I mentioned, could be dominant or recessive. They could be autosomal, meaning they are on chromosomes 1 through 22, or they could even be sex-linked on either the X or Y chromosome. So it's possible for them to be recessive or dominant, and we could track either of those cases using a pedigree. I'm actually going to stop there, and we'll talk about some examples of the recessive and dominant disorders in class altogether. We'll also talk about how they do testing and figure out this kind of information. Okay, but what you know at this point in time is how to solve Punnett squares, test cross, dihybrid crosses, apply the rules of probability, and now you know how to solve pedigrees as well. The best way, honestly, to learn pedigrees better is to do some practice. So that's exactly what you'll do, and that is the final page of your genetics work packet. So please, at this time, review your notes. If you have any questions or something didn't make sense, please write some question marks in your notes so that I can make sure we address those in class. We will certainly review this information together and make sure we have a good understanding. And then we'll talk about some specific diseases and disorders that these tools might be especially useful for. Okay, so that's all I have for you today. And thank you. And if you have questions, please write them down. You can send me an email and we will review all in class when we return from break. All right, thanks, you guys.